Peace and power. Give me 1 second, okay? I can't eat no dry salad, okay? 1 second. Okay, I am back. I had to go get me some more salad dressing. Honey, I don't mind eating salad. I don't need my dressing to be present. All right, so tonight we're going to get more into ooh, the mitochondria. I got a video already pulled up. We're going to watch that. Let's see what it said. Right here. Boom. Boom.
Today, we're going to pump you up.
Okay. I don't know what he's talking about. We're going to show the screen. Boom.
The PR for Powerhouse anywhere in the world, DM us. That whole Powerhouse nickname came from the mitochondria's energy production capabilities, but it's so much more than that. Today, we'll go over how this little powerhouse powers our cells, where it came from, and some new research that you probably didn't hear about back in your middle school textbook.
Okay.
You may remember some variation of this diagram. A classic animal cell with a little jelly bean-shaped organelle called the mitochondrion. Mitochondrion for singular, mitochondria for plural. That diagram is fine when you're learning the structures for the first time, but of course, a real-life cell is more complex. That little jelly bean shape is only one of the possible shapes mitochondria can take. What it looks like can be different from cell to cell. Plus, you don't just have a handful of mitochondria per cell, you have hundreds to thousands of them floating in your cells. And even that number depends on what type of tissue we're talking about. Like a skeletal muscle cell might be 3 to 8% mitochondria by volume, but a liver cell could be about 20%. Meanwhile, heart muscle cells are laughing at those numbers because they're about 35 to 40% mitochondria by volume. They win by a long shot. Now, all mitochondria do have some structural things in common. They each have two membranes, one outer layer, one inner layer, and some space in between them. That outer membrane works like a protective but permeable layer, letting different compounds in or out of the mitochondria. Meanwhile, the inner membrane is where some important biology happens to manufacture ATP. This is the molecule that fuels our major biological processes, so it's often called energy currency. We're going to get more in depth on that whole ATP thing in the next video, but this inner layer, as well as the matrix within the mitochondria, is where the cells generate most of their ATP. Zooming back out to the mitochondrion as a whole, it looks almost like a separate cell in its own right. That's because at one point it was. The most widely accepted theory of how we got these little guys is the endosymbiosis theory. Endo meaning into, symbiosis meaning living together. This word meaning that one cell engulfed another cell and it resulted in a mutually beneficial relationship. About 3.8 billion years ago, Earth's atmosphere didn't have oxygen in it. And the only things living on our planet were single-celled organisms that were anaerobic, meaning they didn't need oxygen to survive. Fast forward about 600 million years, and photosynthetic bacteria were everywhere, taking sunlight and a few other ingredients and cranking out oxygen as a byproduct. A few hundred million years later, those photosynthetic bacteria had produced so much oxygen that it fundamentally changed the composition of Earth's atmosphere. Here's the thing. Oxygen was actually toxic to those anaerobic cells. It's so bizarre that something we need on a daily basis was so deadly back then. It's like if I found out that my ancestors were allergic to tacos. That meant that these anaerobic bacteria were at a huge disadvantage once the atmosphere was made of, what was to them, poison gas. By 2.5 billion years ago, a new type of bacteria started showing up in the fossil record. These bacteria were aerobic, meaning they could use oxygen, and it even helped them create energy. That is an excellent evolutionary advantage when the atmosphere is made of a gas you can use. The theory suggests that eventually, one of those anaerobic single-celled organisms consumed an aerobic purple bacteria that survived being eaten, and they kicked off a symbiotic relationship. That was the first mitochondria. That purple bacteria could consume and metabolize oxygen, which provided energy for the host cell. And in return, the host cell protected the bacteria. We still don't totally know the conditions around that moment of symbiosis, but we have fossil evidence of it starting about 1.5 billion years ago. The result of this ancient endosymbiosis is today's powerhouse of the cell. We kept mitochondria around to make energy for our cells. And since they were once their own separate organisms, they retained certain features of their past selves, one of which was their genetic information. Just like our larger cells, mitochondria need certain proteins to do their jobs. So they need genes to tell them what proteins to make. Our DNA, which makes up our genes, is kept in our cell's nucleus, what I'll call nuclear DNA for the rest of the episode. Some of our nuclear DNA makes proteins for the mitochondria, then ships them out for it to use. But the mitochondria also has its own DNA, separate from the DNA in your cell's nucleus. Plus, it has the cellular machinery to make new mitochondrial proteins, again, separate from the rest of your cell. This is the mitochondrial genome, or the entirety of its genetic information, and it's much smaller than the genome in the cell's nucleus. It's only a small circle with about 16,000 base pairs, while the nuclear genome has billions of base pairs. Now, the vast majority of proteins that get used by the mitochondria come from nuclear DNA, but that mitochondrial DNA lets us make some cool observations. Thanks to sexual reproduction, humans are genetic mishmashes of our parents. So you might expect that our mitochondrial DNA comes from our parents too. As a matter of fact, for a few reasons, we only inherit mitochondrial DNA from our mothers. When you were first developing in utero, most of the chromosomes from your biological parents recombine to form your chromosomes. This is part of what makes you physically different than your parents. But mitochondrial DNA, as well as the Y chromosome from your father, don't recombine, so they get used to study lineage. This kind of DNA does mutate, but it's otherwise well conserved. So the information in our mitochondria's genes are similar to our maternal ancestors way, way back in the past. Sequencing that mitochondrial DNA and comparing genomes has allowed researchers to trace people back to a single female ancestor in Africa thousands of years ago and follow human migration. Now, why does mitochondrial DNA only come from your mother? Good question. The first is that egg cells hold way more mitochondrial DNA than sperm cells. It's around 200,000 molecules in an egg cell and like single digits in sperm cells. Some estimates are a little higher, but the point remains, egg cells outnumber sperm by a lot when it comes to mitochondrial DNA. Plus, sperms store most of their mitochondria in their metabolically active tails. It does take a lot of energy to swim after all. Now, aside from helping our cells make energy and providing clues about our ancestry, ongoing research is showing us some new features of our mitochondria. For example, research by scientists at the Salk Institute showed that mitochondria can kick off a series of events that signal the rest of the cell that it's under stress, the kind of chemical stress that can damage DNA. This phenomenon caught their attention when they observed how defective mitochondrial DNA caused the cell to eject the damaged mitochondria and actually send out a chemical warning signal that strengthens the cell's defenses. So they investigated what would happen if any of that DNA spilled out of the mitochondria and into the liquid around it. When they did, they saw that a certain set of genes were activated that usually activate when there's an invading virus. Awesome, that's exactly what we want our immune systems to do. Attack a virus when it detects one. Now, that same set of genes is also activated by chemotherapy-resistant cancer cells, specifically cancer that's resistant to doxorubicin, a chemotherapy drug that attacks nuclear DNA. When they studied this drug more closely, they found that it caused the release of mitochondrial DNA from the mitochondria, which activated a subset of those protective genes, which then protected the nuclear DNA. The point of this drug was to attack nuclear DNA, but when these genes were activated, it set up a pathway to defend the nuclear DNA, which explains why some cancers were resistant to this drug. The researchers took cancer cells and induced stress on their mitochondrial DNA, and as expected, they activated more of those genes and developed a resistance to doxorubicin afterwards. This research doesn't show that doxorubicin is a useless chemotherapy drug. It just explains why some cancers develop resistance to the drug. They think the purpose of that response is to protect the DNA in the cell's nucleus, making the mitochondria a warning signal that something bad is happening. They hope that if they can find a way to protect the mitochondrial DNA, they'll prevent that immune response within the cell and find more effective chemo treatments. So not only is our powerhouse of the cell effective in generating energy, it's got a fascinating backstory with clues to our past and to our future medical treatments. If you're wondering why we kind of glossed over the energy-generating aspects of the mitochondria, it's because we're saving it for later. Check out the next episode in the series to learn about how our cells generate energy. Yep, I'm going to try to teach y'all how ATP...
So, we are suffocating on a cellular level. This is so deep.
My name is Ariel, and I just started my first job as a project manager. When I was a student...
Thank you.
Dr. Urban here. I want to discuss with you a new and exciting way for you or all of us to revitalize the body and get more energy. New research tells us we can repair
Like
mitochondria and even create new mitochondria through a process called Mitochondrial Biogenesis. Sounds complicated. It's not, but it is exciting. Mitochondria are energy packets inside our cells. Think of mitochondria as little power plants dispersed throughout each cell. Each cell has hundreds of mitochondria. It just so happens that mitochondria have their own DNA. But there's a problem with mitochondria. Although we start out with hundreds of healthy mitochondria, mitochondria degenerate, fragment, and disappear as we grow older. Until recently, only severe starvation and long endurance exercises could create new mitochondria. Otherwise, mitochondria will progress.
So that means, y'all know I'm eating my salad. That means that's why people fast. That's why our ancestors fasted.
Progressively disappear over time. Suddenly, recently, we found an additional way to create new mitochondria using a natural plant-based nutrient or supplement. With new mitochondria, our cells are re-energized, particularly the heart and body. As we boost brain power and slow down the aging process in a way that is light years ahead of simply taking CoQ10, the most common way to address the issue now. However, CoQ10 does not create new mitochondria. Mitochondria are like the jet engines that power airplanes. In this case, mitochondria are the power packets. You may be familiar with the nutrient CoQ10, which our body makes, but less so after the age of 40. CoQ10 is vital in energizing your cells, but CoQ10 is only the energy fuel. Now we know that taking CoQ10 is not enough for cellular energy production. As beneficial as CoQ10 is, CoQ10 cannot create new power generators, also known as mitochondria. Did you know that by age 67, 80% of cellular mitochondria are just gone, and many of the remaining mitochondria are internally fragmented? Importantly, and again, this has nothing to do with CoQ10, which is still important. And likewise, CoQ10 diminishes as we age. Remember that CoQ10 is the jet cellular fuel, while mitochondria are the jet engines that use the fuel. Everything changed when the plant nutrient PQQ was discovered. PQQ stands for pyrroloquinoline quinone. PQQ is 5,000 times more efficient in sustaining cellular energy production. Your brain is the most energy-hungry organ in your body and contains the most mitochondria. You can see where this is going with regard to brain energy and increasing it. Once mitochondria are destabilized, that particular cell goes into a death spiral. PQQ stimulates nerve growth factor, which triggers the growth of nerve cells in your brain for sharper and clearer thinking. PQQ protects against memory-robbing plaques. Think Alzheimer's dementia. Today's best PQQ can be found combined with a unique form of CoQ10. The special CoQ10-H2 is 8 times more powerful than standard CoQ10. How about that? This makes other forms of CoQ10 obsolete. Again, this new form of CoQ10 is called CoQ10-H2. It was through my need for a stronger heart while taking statins that I discovered the work of Dr. Al Sears, MD. Dr. Sears is a noted researcher and clinician who has a supplement which puts together or combines the latest CoQ10-H2 with PQQ, adding special vitamins plus the immortality vine. I just want you to remember that there is more than PQQ in Dr. Sears Ultra Excel 2, that special PQQ supplement. Ultra Excel 2 combines the best ingredients known to maximize energy production inside the cells. I know I've thrown a lot at you, so let me summarize. CoQ10 is good and necessary. You should be taking it. However, you should be taking CoQ10 as CoQ10-H2. CoQ10 is mandatory in some countries, for example, Japan, if you take statins which interfere with CoQ10.
Wow.
That's another issue for another video. The natural plant nutrient PQQ is available to repair and regenerate new mitochondria. It's exciting. By combining CoQ10-H2 with the best PQQ, you have the best combination of ingredients.
Listen. This stuff is also in relation to Alzheimer's, diabetes, autoimmune disease. Why aren't they teaching this? What's this about? And then they saying if you combine HIT with it,
No medical exams on policies under 1 million.
It helps even more.
PrevMed, heart attack, stroke, cancer prevention, disability prevention. Prevention sometimes takes some discipline in terms of lifestyle, but it's a heck of a lot better than getting a bypass graft or becoming senile. This is an image of what happens with the mitochondrial aging process and the impact of decreased mitochondrial function on aging. That's called the Mitochondrial Theory. Why are we talking about that again today? This is, it may sound like science, but actually we're talking about a very practical thing. High intensity intervals, HIIT, high intensity interval training. And we're talking about this not just for young people, we're talking about it for older people as well. 65 to 80-year-olds. We're going to talk about a study where there were two groups, 18 to 30 and 65 to 80, and they put them through several months of interval training and looked at heart, lung, circulation, performance, also mitochondrial performance. So let's go back then and talk about the mitochondria. Again, this is an image of the mitochondrial process of aging. This first step is damage of the DNA in the nucleus. Well, I mean, if you look at Walter Longo and the mimicked fasting activities, his point would be it's not even that the DNA is damaged, it's just that it's not being transcribed. I think David Sinclair would talk about lack of transcription of some controlling genes as well. If you look at this second stage, so the first stage is DNA, whether it's DNA damage or lack of transcription driven by some controlling genes, they still start hitting a lot of the same pathways, the pathways used in respiration, and respiration, the center of respiration is the mitochondria. Again, you'll see some names that we mentioned in other videos. SIRT1 or the sirtuins being researched by David Sinclair at Harvard. AMPK, these are impacted by metformin. And again, all help the cell take food and turn it into energy. So if these metabolic pathways are damaged, then basically you're getting damage in performance of the mitochondria. And the mitochondria are the powerhouse of the cell. So again, this last image shows you get damaged mitochondria, you get damaged neurodegenerative damage, in other words, senility. You get damage to the heart, the lungs, basically aging tissue. And so the human goes from peak health in adult to poorer health as an older individual. So who did this research? His name is Srikumarian Nair. Where is he? He's a faculty member at Mayo Clinic in Rochester. He is a cellular biologist who's been doing a lot of work on cellular metabolism, aging, mitochondria, again, all of these areas that we've just been talking about. So what did he do? As I said, he took two groups of people. One group was 18 to 30 years old. The other group was 65 to 80 years old. He had them doing high-intensity interval training for, I believe it was 3 months. And then he did a few things. He looked at performance of the heart, lungs. He actually did biopsies of the muscles and looked at the mitochondria. So this is an unusual study in that he actually did muscle biopsy. You don't see a lot of studies where they've done that. And what that does is it creates a lot of value for the study, because now we can start looking at the cells, looking on an intracellular level at the mitochondria to see what the impact was. So here's what we saw. or here's what they saw. On the youngsters, 18 to 30, you had a 28% improvement in heart, lung, and circulation health parameters.
Circulation.
In the older folks, 65 to 80, you had a 17% improvement. What about the muscle biopsies in the mitochondrial function? 69% improvement of mitochondrial function in the older folks, and 49% improvement in the younger group. So the younger group had mitochondria that were already healthier to start with, didn't have quite as much improvement that they could experience, which is exactly what you'd expect. Now, when you see some reviews of the study, one of the reviews said, look, well, it didn't make them younger. And in fact, if you did high intensity intervals, but you're eating donuts all day long, you're still going to be in bad shape. I got it. I think we all understand that. But again, this was a great study in that it actually showed improvement of mitochondrial function using muscle biopsies. Now, so a lot of my patients are 65 to 80 years old, and they're folks that are basically just doing 1 or 2 miles of walking 3 or 4 times a week for their extra exercise. How can they get a high intensity interval? They can walk up hills. They can walk carrying a weight vest. They can use a bicycle ergometer, indoor ergometer, or an elliptical trainer. So how do you measure? You can measure it with pulse. You can measure it with increased breathing. What's the impact of high intensity intervals on insulin resistance? The number one cause of cardiovascular inflammation. It is the number one exercise that improves insulin resistance. So again, and maybe it's because of this improvement on the mitochondrial function. Ka-ching. So what about interval training for younger people? Again, you can use similar things. You can do it with running. Professional cyclists have a very simple type of high-intensity interval training. Very many of them use a 45-second high-intensity interval with a 30-second recovery phase. So again, 2 phases to an interval. The high-intensity phase, where you're going, it needs to be high intensity. Pulse well over 100. For some of these younger athletes in the 140s, 150s, even some of the older athletes can achieve that, but they have to be in very, very good shape compared to most folks in the 50s or 60s. So again, back to the cyclists and what a lot of them use: 45-second high intensity phase, 30-second low intensity phase. That's one interval. Do a warm up, do 5 to 10 of those intervals, and then do a 5-minute cool down. You're talking about 20 to 30 minutes total for an exercise. You don't need 3 hours in the gym to do these things. Now, are there other ways to do this? Again, yes, you can run. You can go up hills. You can do what are called hill intervals. And each of these has a slightly different component. I saw an ad once that said, "It isn't that diabetes runs in my family, it's that nobody in my family runs." I get it.
I've always stated that none of this stuff is hereditary. They just tell you that to have you in a state of fight or flight. We'll look at the TED Med because they tell a lot of truth in TED Talk. And the numbers for this video was 5-5. It was 1-4-4-1. And so we know they like their numbers too. But yeah, none of this stuff is hereditary. Don't listen to colonizers about your ancestral foods and dishes.
It could be argued that every time somebody decides to have a baby, they're engaging in a game of genetic Russian roulette. Now, we all start our lives like this, a single cell, the fertilized egg. At this stage, the odds are overwhelmingly stacked against us. Between 60 and 70% of these eggs never develop to a stage where they can form a healthy baby. So, in this sense, we're all survivors. Well done. Now, whether this egg goes on to form a healthy baby depends largely on the DNA it inherits from its parents. And it's got 3 different sets of DNA. It's got 2 nuclear genomes, packaged into chromosomes, and contained in large structures known as pronuclei, which you see here. Now, the female pronucleus contains the nuclear DNA we inherit from our mothers and is transmitted by the egg. The male pronucleus contains the nuclear DNA we inherit from our fathers that's transmitted by the sperm. And when this fertilized egg begins to divide to form a 2-cell embryo, these 2 genomes join forces to confer on each of us our unique genetic identities. So what about the 3rd genome? The 3rd genome in the fertilized egg is known as the mitochondrial genome. This we inherit only from our mothers. The mitochondrial DNA is contained in the mitochondria which are scattered throughout the egg. It consists of a tiny circular molecule, of which human eggs contain about 300,000 copies. Now, the sperm also contains some mitochondrial DNA, but this is destroyed during early development. So the main job of the mitochondria is to produce the energy our cells need to sustain life. And the genes encoded by the mitochondrial DNA are essential for this purpose. Harmful mutations in these genes cause a broad spectrum of debilitating and fatal conditions, collectively known as mitochondrial DNA disease. Devastating diseases for which we currently have no cures. So for children who inherit mitochondrial DNA mutations, disease symptoms may not become evident until weeks, months, or even decades after birth. And sadly, mitochondrial DNA disease frequently goes undetected until the affected child or adult develops muscle weakness, brain seizures, or heart failure. In the worst cases, children die before reaching the age of 5. Now, women who carry mitochondrial DNA mutations are generally fertile. Yet for them, the decision to have a baby is truly like engaging in genetic Russian roulette. The baby may be perfectly healthy, or it may be seriously affected. There is no way of knowing. The risk of transmission is unpredictable, and this makes for profoundly difficult reproductive decisions. And what makes it doubly tragic is that in many cases, women unknowingly pass these diseases to their children. The first they learn of their own mutation is after their child is diagnosed. So naturally the question arises, isn't there anything can be done to help these affected families? Isn't there some way we can detect the problem early, or better still, fix it before the baby is born? And the answer is yes. Our work is making it possible to replace defective mitochondria with healthy mitochondria so that children born to affected women do not develop disease. We are intervening not just before babies are born, but before they're even conceived, so that affected families no longer have to make these near impossible decisions about whether or not to have their own children. Now, it's been a long road to get to this point, and as you might imagine, we've had many scientific challenges along the way, but we've also, of course, there were ethical considerations, and we've also encountered legal hurdles. And in order to give you a feel for the magnitude of the task, it's important that we understand some of the basics of mitochondrial biology and human reproduction. So you might ask, how do the mitochondria come to have their own DNA in the first place? Well, our mitochondria descend from ancient free-living bacteria who about 2 billion years ago were engulfed by precursors of our modern-day cells. And this has been referred to as the most important meal in history, because it turned out that the mitochondria could use oxygen to generate energy from the food we eat and to store it in the form of ATP. ATP is an energy-carrying molecule which provides the fuel for life in all living things. So during evolution, the mitochondria have outsourced many of their genes to the nuclear genome. But for reasons that we don't fully understand, they have retained a tiny remnant of that tiny circular molecule we saw earlier. It consists of just 37 genes and encoding only 13 proteins. Now, the mitochondrial DNA may be small, but it is very important. These 13 proteins are essential for energy production, and mutations that affect their function limit the amount of energy available to the cell. And this is particularly problematic for organs such as the brain and the heart, which consume enormous amounts of energy. But it's not, while the brain and the heart are often the first to be affected, mitochondrial DNA disease is notoriously diverse in its manifestations. In essence, any organ can be affected at any age. And disease-causing mutations in the mitochondrial DNA are surprisingly common in the human population. They have been detected in 1 in every 200 babies born. Now, fortunately, they're present mostly at very low levels, but for about 1 in every 5,000 babies, the mutation load is high enough to cause disease symptoms. So this brings me to a complicating factor in the inheritance of mitochondrial DNA mutations, or mitochondrial DNA disease. Mutated mitochondria coexists with normal mitochondrial DNA. And it is the relative levels of these two that determine the severity of disease symptoms. And here's the problem. Women who carry a mix of normal and mutated mitochondrial DNA produce eggs with widely varying mutation loads. This is what makes it so difficult to predict the risk of disease in their children. This is what makes it a game of genetic Russian roulette. So working as I do in the field of clinical embryology, I've had the great privilege of spending much of my working life observing the very earliest stages of human development. And during the first few days of life, the embryo undergoes successive rounds of cell division. And then at about 5 days after fertilization, it forms this beautiful structure known as a blastocyst, and at this stage it's ready to implant on the wall of the uterus and hopefully give rise to a viable pregnancy. So one option currently available to women who carry mitochondrial DNA mutations is to take cells from these very early embryos and to test them so that we can identify embryos with the lowest mutation load. This provides a way of reducing the risk of severe disease symptoms in a child. However, it doesn't work for women who consistently produce eggs with high mutation loads. In other words, current treatments are not suitable for those at highest risk of transmitting serious disease to their children. So what can be done to help these women to have healthy babies? Now, in theory, the problem could be solved by scooping up all of these defective mitochondria, but there are so many of them, and they're scattered around throughout the egg, that this would be technically impossible. So an alternative strategy is to rescue the nuclear genome from its defective mitochondrial environment. And by this, I mean to transplant the nuclear genome from the egg of an affected woman to the egg of a donor who does not carry the mutation.
Now, I just want to stop her and say, we know how these people operate. And so what they do is go about trying to find a scientific way to sort through who they think is worthy. If you could separate it, the way she's saying is, with the mutation, a mutation could be good or bad. It's not just what they deem bad. Let's say, for instance, the mutation is healing, rapid healing. And so why would you want to separate it? They don't know what the mutation is. They're just going by a genome or a number, so to speak. So chew the meat, spit out the bones, because we know they like to play God, and they don't speak half truths. These people are trying to tell you that it's playing Russian roulette having a child. We've been having children for generations, okay? Yeah. So their numbers are down, and now they want to tell you, oh, it's Russian roulette. Nah.
Nice idea, but its execution is far from trivial. For a start, the human egg is no more than the diameter of human hair, and to transplant its nuclear genome would require microsurgical techniques, which would need to be performed under sterile conditions while keeping the egg happy. Now, before we could even attempt to do this, we had to obtain a license from the UK regulator, the Human Fertilisation and Embryology Authority. And believe me, that was not an easy process. It took the regulator more than a year to satisfy itself that the experiments we proposed to do were permitted under UK legislation as it was at that time. And we were awarded our first research license in 2005. Once we got the go-ahead, our starting point was to do proof of concept experiments using abnormally fertilized eggs. Now, abnormally fertilized eggs are generated during routine IVF treatment, and they generally arise as a result of the egg being fertilized by more than one sperm. And they're not suitable for use in treatment, and couples willingly donate them for research. So using these abnormally fertilized eggs, we were able to demonstrate that it was technically feasible to transplant the pronuclei between human eggs. So having done that, the next step was to see whether this pronuclear transfer could provide a reliable and effective treatment to prevent mitochondrial DNA disease. And for this, we needed a supply of normally fertilized eggs. And we owe a huge debt of gratitude to the women who so generously donated their eggs for this project. Without them, we could have made no progress. And it must be said that that progress was indeed slow. The first major setback came when we realized that the techniques we had so painstakingly developed during the proof of concept phase simply did not work very well for normally fertilized eggs. 40% of them did not survive the procedure, and this was really problematic because we knew that with such heavy losses, the pronuclear transfer could never make an effective clinical treatment.
That's the
so
Russian
we had to go right back to the drawing board, and we tried a number of things and finally solved the problem by radically changing the timing. So instead of waiting until the next day after fertilization, we transplanted the pronuclei as soon as they became visible. And this allowed more time for the egg to recover from the microsurgery before having to embark on the complex choreography required to divide to the true cell stage. I mean, the change of timing seems perfectly obvious now, but it wasn't so obvious at the time, and perhaps this was because it meant performing microsurgery late into the night and often into the early hours of the morning. Unfortunately, the strategy paid off. More than 90% of eggs survived this modified procedure. And here you can see it here in this video. You can see this egg has been fertilized about 8 hours beforehand, and the pronuclei are being removed one at a time from here. And then they would be placed back under the outer shell of another egg from which the pronuclei have just been removed. And these will fuse very quickly to form a reconstructed embryo, which in the context of clinical treatment would contain the nuclear DNA from the affected woman and her partner, but the mitochondrial DNA from an unaffected donor. Now, we had numerous other challenges to overcome before we became confident that this pronuclear transfer technique could provide a safe and effective treatment to prevent a transmission of mitochondrial DNA disease. Meanwhile, back in the outside world, our work was subject to unprecedented level of public interest. There were public consultations, there were legal debates and ethical debates, and intense media interest in what had by then become known as three-parent babies or three-parent IVF. This term is, of course, very misleading because while the woman who donates her mitochondrial DNA gives the great gift of health, her mitochondrial DNA does not encode any of the characteristics we would consider to confer on us our unique genetic identity. And moreover, in the UK, at least according to UK law, this donor has no legal responsibility for the child. Notwithstanding this dubious lexicon, the UK Parliament voted to change the law to permit the use of pronuclear transfer in clinical treatments to prevent transmission of serious mitochondrial disease. That happened in February 2015, and then earlier this year, in March 2017, we were granted our first treatment license, exactly 12 years after we were granted our first research license. So what do these treatments mean for affected families? Obviously, for women of childbearing age, it gives them the possibility to have a genetically related child with a greatly reduced risk of transmitting disease. But it goes beyond that. And a very poignant moment for me was...
Now, y'all can see this is TED Med. Y'all can go over and look at this. a groundbreaking way to stop mitochondrial disease. I'm going to pull up one more video on what mitochondrial disease is, but I want to point out that these people be lying. And so it's like you're saying, and this is what the scrutiny was, them trying to genetically create a child with no defects. Again, what is a defect? Because it could be a heightened gift depending on who's looking at it, right? A lot of these diseases, they don't know anything about them. They assume, and they also practice medicine, okay? And so with that being said, it's like you're going to take the egg from the mother who you say may, pass this to the baby, or she may not. What if the cells, because we're talking about ancestral cells that were around when carbon dioxide or oxygen was poison to us. Now we get the oxygen from the trees, but we know that oxygen from the trees is tree waste. We're breathing in waste, right? And so they do all these experiments, and I truly believe, my own belief is that when you go the route, and I know men and women go this route, I just don't believe the soul is present, because you create life with that spark, and then if they're injecting it with a needle, there's no combustion, no spark to create that life. And we all know that the spark is the soul. And so be watchful. Like I said, chew the meat and spit out the bones.
Was meeting a woman of about my own age, who was at that time.
Oh, one more thing. I had forgot. That's what I was thinking about, the age thing. They tell us that the older you are, you're high risk for pregnancy. What I've noticed in my experience, the older you are, the more mature and knowledgeable your children are. Your cells are at their best. They've matured. I've had friends 14 have children, and their children end up being just this wild, chaotic being. And then I have people that wait till they're 40. Most of my family members wait till they're 40 something, and the children are either geniuses or borderline insane. And we know borderline insanity is borderline genius. And so we have to watch the narratives that they push.
Succumbing to the effects of her own mitochondrial DNA mutation, a mutation she did not even realize she had until her brother became seriously ill. And
they keep talking about illness, but there's a pro and a con to everything. What's the plus?
We know that mitochondria may indeed be endosymbiotes, in the sense that millions of years ago, these tiny bacteria were engulfed by larger cells and ultimately evolved to be codependent with our cells. We provide them with foodstuffs and protection from predation, and in turn they provide us with the majority of the ATP energy currency that our cells need to do work. What you may not realize, however, is that each mitochondrion has its own DNA, its own genome. So what that means is that while we are accustomed to thinking of our cells and their genomes in terms of what exists in the nucleus, all of the genes that we think of as the ones we inherit from our mother and from our father, we cannot forget that the mitochondria in every single cell also carry their own DNA genomes. Now, where did those DNA genomes come from? Well, another interesting fact is that
Now I want to stop and just point out that the old science drew the pictures of the mitochondria in that little bubble and all that, but the new images of the mitochondria, they look like roots, like that of a plant, right? And so the mitochondrial DNA, the mitochondria has its own DNA. Let's start there, but It's the root of the cell. It all goes back to the beginning. Okay?
We only get mitochondria from our mothers. The mitochondria come with the egg, while the sperm that we got from our fathers, in terms of fertilization, do not contribute any mitochondria to what will ultimately develop in embryogenesis into us, into an adult human being. So this is, of course, a debt that we owe our mothers. They are the source of our mitochondria. But then if you want to think about what we inherit, and are there possible mutations that we might inherit, we can't just think about what we inherit in terms of the nuclear genomes, what we inherit from our father and from our mother. We also must think about what mitochondria we inherited from our mothers. It was fairly recently when we discovered that there are a number of human diseases, usually metabolic diseases, that are tied to the inheritance of mutations in the mitochondrial genome. Many of these diseases, as you might expect, are related to lower than normal levels of ATP or defects, for example, in the electron transport chain, defects that really affect, if you will, the energy metabolism that we all need so crucially for all of life's processes. So the question is, where did these defects come from in mitochondria, and how can we understand the process of inheritance that occurs? Let's begin by thinking about a cell's mitochondria and what the makeup of those mitochondria might look like. So if we have a cell from our bodies, we have a nucleus, and then we may have multiple mitochondria in a cell. Usually we'll have anywhere from 10 to 20 mitochondria. Now, when we think about this, we generally think of the nuclei, or a single nucleus in a single cell, as having a particular genome. But what we don't usually think about is that the mitochondria in a cell are in fact a population. They are not all the same. And in particular, if you think about their genomes may not be all the same. So here we're shown a number of mitochondria, and we're distinguishing their genomes by color. So we have, let's say, 4 mitochondria that I'm designated as having one particular genome in purple. We have 2 in pink, and we have 3 in orange. Now let's suppose that there's a defect in the mitochondrial genomes, and the defect is associated with these 2 mitochondria that are pink. What this means is that the defects are associated with 2 mitochondria, but we have another 4, in fact 7 mitochondria that are otherwise perfectly healthy. And so when it comes to the state of that cell, whether or not there's a negative impact of those mutations will depend on how many mitochondria are defective. Mitochondrial diseases are really quite devastating. If you think about it, it's not really surprising, because given the role that mitochondria play in providing the majority of ATP that we need, any disease that disrupts their ability to produce ATP, to manufacture ATP, can have significant metabolic impacts. So mitochondrial disease often involves, for example, muscle weakness and wasting, because muscles use a significant amount of ATP. It also can involve cognitive defects, sort of degeneration of the brain, because as one would expect, neurons and nerve cells in the brain also utilize an enormous amount of ATP. How common are mitochondrial diseases? They're really one of the more common kinds of genetic diseases. For example, in the United States, where you'll see a frequency of roughly 1 in 5 to 10,000 births might have some kind of mitochondrial disease. Because of the severity or the possible severity of mitochondrial disease, you also frequently see lethality by age 2. So that quite often, if there's a severe expression of a mitochondrial disease, you might have a stillborn kind of situation, or you may also have a case where the child dies by age 2. So clearly, this is a very severe kind of disease, but it also reveals a remarkably different kind of inheritance than anything we've seen before.
Mm. Okay. So, this is just interesting to me. So what I do when I get interested in something, I just dig and dig and dig. And so this is part 3. I don't know if we're gonna do any more on this topic, but you get it. We went over the supplements that you can take for mitochondrial regeneration and also to create new mitochondria. That supplement will be the PQQ. Also, the NAD is a supplement that helps with weight loss. It's also in relation to the CERT food diet plan, and that's NAD. And the other main thing is CoQ10. They also name alpha lipoic acid, acetyl L-carnitine, NMN, phospholipids, and D-ribose. These are 7 super supplements that will help your body to regenerate and create new mitochondria. It's too many of us walking around on a day-to-day basis exhausted. I hear so many people saying it's male and female, and so the conclusion I've come to is our cells are not cleansing that waste out. I'm sorry, I'm looking at my vision board like, what is happening? But anyway, I will be on my supplementation. I've been working out a year. I thought my energy would be up by now. It's okay, but I'm going to try this out to see. I've also been adding a lot of vegetables in my meals on a daily. So this will just be one more step to longevity. A lot of the people that take the PQQ said that it's the fountain of youth. They can feel the difference within 4 hours. Not 4 weeks, not 4 days, 4 hours of taking the supplement. I've researched several supplements. I don't recommend any at this point, but when I do get some, by then I'll be drop the link in the description box. But please look into this. If you're one of the people that are exhausted, even though you're getting sleep, but you're still feeling exhausted, your mitochondria might be very low. You just never know. We know when you have a baby, they test for all these defects, but they never tell you in specific what they're testing for. They just say defects. Make sure they don't have this is that. And God forbid, I don't know what they do with the blood. They stabbing your baby foot. We know they stealing your baby rights right out the womb. But they never teach you about your body. Everyone's body starts with a cell. If you're talking about getting a root of things, diet is way down the list. And I was just telling Tanisha, no matter what you eat, a lot of people subscribe to all these different diets. I always bring up we are the only culture that allows foreigners, colonizers, to call our ancestral dishes slave foods. We are also the only culture to where our ancestors are living past 100. So it was not the food. My grandparents ate the same way their whole lives. They both lived to see over 100. You got to go deeper than that. They selling you all these fads and these diets because they want to charge you more. We know the organic and what else are they naming it? Organic, no put all this stuff they label while they're poisoning you. They get to charge more for it, and that's just that. They are charging you more because they're telling you it's healthy, and it's not making a difference. It's not. That's all I got. It's just this right here is one of the downloads that I got during my fast, and so I'm gonna keep digging. Like I said, I don't know if I'll get some more information in regards to mitochondria. We're just gonna keep digging, but you got to get to the root of the problem. if the mitochondria is the roots of the cell, and the cell is your soil, the membrane of the cell is dry, depleted, how can that tree live on? And so that's why the death rate or the age, the decline in death, living a long time, longevity is down. It's because we're not studying the root of the problem. It's the cell. You got to get down to the cell. What do they teach you about the cell in school? Absolutely the bare minimum. They still teaching and using the old cell model, and they've come to realize that the cell is not even shaped like that. And so I hope this puts you on your journey. Thank you for tuning in and just tapping into the energy. I got to get back to creating content. This is what I love. I love knowledge and sharing. And so we're going to get back to the root. This is how my channel got started. Wish everybody well. And we do have to grow out. That's why everybody who pushing something. It's just when I hear people talking and they talking about we supposed to eat this and we supposed to eat that, and they not growing any food or don't own any animals, I just let them talk. They like to blow hot air. If you're not growing your food, I don't want to hear it, And that's just me. I don't follow no fads. I do what grandma did. Grandma lived a long time, and so that's what I'm at with it. But yes, go on your own, because we know that in the first video, I showed y'all where they are taking the corona patients or the vaccine recipients and melting them down with lye, turning them into a bio-waste sludge, and they're spraying this sludge on the crops, okay? There's no getting around it. If you're not growing your own food, you're eating that sludge, which is in essence humans. They're growing plants for humans. And so, yeah, go figure. Yeah, let me get out of here. It's 1. I gotta get some rest. I stayed up to 4 last night doing this video, this part 2. And so I'm going to bed tonight. Peace and power, everyone. Thank you again for all your continued support. And I'm going to get out of here.